We're very excited to have her, and we look forward to hearing your talk, Olga.
Great. Am I on now? Awesome. Cool. Thank you so much. Ooh, someone's lift just came here. Sweet. So I thank you very much for that really kind introduction. I'm really excited to be here and tell you a bit about where I work, a bit about what I've been doing since I've been there. So a bit about me to show a bit of personality. So as Josh mentioned, I double majored in math and biological engineering. I also play cello. I have also become famous for defending my PhD in the quinceañera dress, which was my trick to get people to actually attend my defense, because you work like super hard for so long and then like 4 people come to your defense. So I had a gimmick, and it was actually pretty well attended, I will say. So you might want to try that trick. And I tried out for the Golden State Warriors dance team, and I'm a member of a hip hop dance company. So outside of science, my life is dance mostly. So let me tell you a little bit about the BioHub. So the Chan Zuckerberg BioHub is an independent research organization, medical research organization, nonprofit 501(c)(3). Our sister organization is the Chan Zuckerberg Initiative, which many people confuse for the BioHub. One of the many projects of CGI is science. So BioHub is one of their projects. They also work on the Human Cell Atlas. They also work on policy in health and education, getting health in K through 12 schooling integrated in Palo Alto. So they do many things. BioHub is just one of many. So we have money for 10 years. We have affiliations between Stanford, UC Berkeley, and UC San Francisco. And we have 97,000 square feet. Most of which is lab space. I can't tell you how much of that is lab space. There's that's supposed to be some impressive number. The goal of the BioHub is to study basic biology. I believe the official stated goal is to cure, manage, and treat all disease by 2100, which is a lofty goal. And if you think about it, you know, we didn't have antibiotics 100 years ago. We also didn't have vaccines for many diseases 100 years ago. We didn't have CAR T therapy for leukemia, for multiple myeloma, for many different cancers, you know, 30 years ago. So change happens quite quickly, and I think if we invest in the basic biological research, we can get closer. The Chan Zuckerberg BioHub has great leadership in our 2 co-presidents, our COO, and this is our scientific steering committee, involved in many different projects, whether they range from the expertise of Jennifer Doudna in genome engineering, and from Peter Kim to infectious disease. In there are 3 main tiers of Chan Zuckerberg BioHub, CZ BioHub. The BioHub Investigator Program. So this is a program through which professors at UCSF, Berkeley, and Stanford, about 50 of them in total, now about 80 for a different grant, have come to BioHub every 2 weeks and talk. And they can collaborate with one another. One of my projects that I won't talk about today is a tick genome assembly project that I'm working with one of the BioHub investigators on. The other aspect is the platform development, where I am. So this includes the data sciences platform for me, microscopy or imaging, genomic sequencing, and engineering such as Fab Lab and genome engineering. Finally, we have the research projects, which include the Infectious Disease Initiative and the Cell Atlas program. So I'll describe them a little bit more in detail. So the Investigator Program, we have administered by Lubert Stryer, who I didn't know was famous until I came here. But about half of them are biomedical. We have quite a bit of physics and math background people as well, or people who are focusing on, like, say, image reconstruction techniques. There was one talk where someone was making a camera that could, with Scotch tape on top of a cell phone, could resolve the image quite nicely because of the irregular the regular impurities in Scotch tape could change the lens, change the image as it came into the lens, and allow you to focus and make an even better image. So we have quite a few investigators. About half of them are young associate professors. There's a lot of them. The BioHub platforms, as I mentioned, are engineering. So this is genome engineering and engineering fab lab, breadboards, lathes, those kinds of things that you need to be really careful around. Genomics, genomic sequencing, imaging, and I'm in data sciences. So the data sciences team at CZ BioHub is just under 20 people. I was the second data scientist hired almost 2 years ago. I was the second data scientist who started. James was technically hired before me, but I started earlier, so we have our beef there. And we have a really great team of people working on a variety of projects, including phylogenomics, phylogenetics, detecting outbreaks in hospitals, outbreak surveillance, working on transcriptomics work, working on deep learning, machine learning, especially these two for imaging and image analysis, being able to do deep learning on the fly while you're imaging something. And these two are interns, so undergraduate students. One of the great things at BioHub is we have someone, Loic Royer, who uses light sheet microscopy to image embryo morphogenesis. This is in a fly embryo, and this is a recording every 30 seconds of the nuclei labeled in a fly, and he's very interested in cell migration as it occurs and how exactly the cells move and migrate throughout development. Really incredible. And it's only through light sheet microscopy, which is less phototoxic to cells because you're only giving a very small amount of light and not illuminating the whole field. And so he is building one of the world's best light sheet microscopes in the world now. So I'll be done with the corporate part soon. Just stay with me. Some of the other internal projects are the Infectious Disease Initiative to detect, respond, treat, and prevent infectious disease. So this is detecting hospital outbreaks, responding to them with medicine, treating them, and preventing them by discovering new vaccines for things we don't have vaccines for, so Zika, dengue, and their interplay. So I mentioned the Chan-Zuckerberg Initiative and didn't talk much about them. There is a engineering team at CGI that's dedicated to the BioHub. So this is the CGIIDC team. They built a tool called IDSeek that allows you to upload sequencing data, FASTQ files from anywhere in the world, and then detect what is inside. So what samples are inside, what microbes, what species, what do you have there? After they do a host depletion, so if you can specify your host, human, mouse, mosquito, and so on. This is widely used by the infectious disease team I mentioned for their California mosquito surveillance program. So you collect mosquitoes from all over California and grind them up and see what's there. The IDSeek is a collaboration and has been used by all around the world, and they've just had grants to train people outside of BioHub all around the world, about 6 groups, I believe, who will come to train at BioHub on making the libraries and making the sequencing data. And then people at BioHub are also going out into the field to outreach and teach how to use the software as well. The Cell Atlas Initiative involves people who are interested in where these proteins are that are in cells. If you look at every single one of them, where does it actually end up? So Loic is behind the video I showed you of Drosophila development and is interested in lineage tracing in worm, fish, and mouse. Spiros is the reason the Tabulomeros project even worked or happened, I would say. Like, it's a big thanks to him. But he is very interested in diversity of cell types using single cell RNA seq in a variety of systems, including cancer, and lung cancer specifically. Greg is hilarious. Like, I don't even know what he's drawing here. But he is a theoretical biologist, the head of the theory group at BioHub. And he and I are working on the tick project, and he has some cool ideas about genome assembly and spin glass. And that's all I know. To me, what makes BioHub unique is that I'm able to have the freedom to do things that I don't think I could necessarily do in a traditional academic or traditional corporate setting. For me, one of the things I like doing is sharing. So I have a live streaming channel called Bioinformatics Beyonce, where I live stream either my real work, so what I'm working on today, like I'm trying to learn TMUX. I've used Screen for years, and now I have to figure out which key mappings to use and how to make multiple panes or whatever. This is me working with a colleague on some orthologous gene stuff. I forget. So either real work, interviews with scientists, or pair programming. So I'll work with someone and help fix up their Python code or whatever they're working on, and we'll talk through it. The other thing I do is I host a weekly cupcakes and coding session, which is a programming demo or tutorial taught by someone typically with 0 to 2 years programming experience. And it's focused on practicality. So I bang my head against the wall for a month, so you don't have to. So please take this code so that you can learn how to make this fax plot in R without me. So you can learn to visualize this in Python. You can intersect your genomic regions and so on. So it's very much focused on practicality. And alternates between that and office hours. So what used to be just demos, and we changed it to include office hours because people still had a lot of questions about what they were working on, trying to implement what they learned last week, and implement that to their own work. Okay, that's the end of the corporate stuff. Do you have any questions about BioHub so far?
Well, we talked about it a little bit, but I mean, maybe you might, it's interesting how it's developing in collaboration with Berkeley, CSF, and how, I know this Pasteur, Dakar, and other places. I mean, as a hub, I mean, is the mission to kind of establish worldwide collaborations in these various areas? I mean, is that really what it's about?
So the question was about establishing worldwide collaborations from BioHub to throughout the world. That is from my understanding, that is one of the goals, is to not just be a place that's in one place that's in San Francisco and that only certain people can live at or attain to, but to be a resource for and collaboration with the whole world.
So you also mentioned that there's not the necessity for the investigators to get grants, that there's funding for internally for that.
Yeah, for group leaders that are staffed within BioHub, there's no need to apply for grants. For the BioHub investigators who are outside of BioHub at UCSF, Berkeley, Stanford, they do the typical grant stuff. There's some weirdness that they can't have the BioHub or CGI grants if they have an HHMI grant as well. So some people pick and choose there.
So you were at the UCSD before, and you know about the academic sector.
Yeah.
So what are you seeing as the difference? Get into an environment like
so
there's not that pressure.
Yeah, I mean, the difference between academic life and BioHub. So I don't think BioHub is a true industry setting, in my opinion. We're not trying to create a product or a deliverable, so like a medicine or a treatment, so to speak, so directly. Like, to me, the output is still research, whether that be in the form of tool development, whether that be in the form of data, in papers, in blog posts. I think there's many outputs of research. So relative to my experience academically, certainly less pressure to publish like a specific paper or like the biggest paper possible with every technique in it. And to me, at least, I've enjoyed a bit of a slower pace for just sitting and thinking and exploring problems. So yeah, I would liken it more to a research institute that's also internally funded. Any other questions? Going once? Yeah? All right, cool. So now I get to talk to you about the research stuff. And I am working towards a phylogeny of cell types. And I will tell you now that this is early work. I've been working on this project mostly by myself for the past 6 months, and I gained people to work on it in the past 2 months. So any feedback is welcome, but know that it's still early. So we'll start with thinking about how we can build a phylogenetic tree of cell types. We'll talk about how most single-cell RNA-seq analyses are limited to annotated genomes, how we can use k-mer sketches to bypass genome annotation, and how if we use protein sequences, we can now compare across species. So I like starting with thinking about the number of cells in the human body, which is about 37 trillion. And all of these have very different shapes, forms, and function. I think if you like really stop to think about how many different kinds of cells there are in the body, it makes you just like wonder at life and how life is possible. And what's very interesting about this is that each of these cells has nearly identical DNA. So what's really making them different? One thing that we can think about for cells is they are an intermediate between the DNA we can sequence and the phenotype that we see. So if there's a particular variant in the DNA, we can see it manifest itself in a differently acting cell type, which can then manifest itself as a disease. So the argument here is that understanding cells is a key step in understanding the genetic underpinnings of disease. And not so recently anymore, single-cell RNA-seq has come to the forefront as a way to identify cell types. So given some unbiased, relatively unbiased group of cells, we can capture them and explode their contents, explode each cell, explode its molecular contents, measure what molecules we were able to capture there, and from these measurements of high and low, we can then group the cells together based on their similarities. And from these similar groupings, we can then identify cells that tend to group together, tend to be of a similar cell type. And this process is still under active research about exactly how to identify these cell types. What I am, okay, I just added, damn, there was a bit of a, okay, that's okay. We'll go back here. So if we think about cells as a fundamental unit of life, right? Cells are a building block of life. After cells, cells come together to compose of tissues, tissues to organs, organs to organ systems, and organ systems to whole organisms in terms of the hierarchy. What I'm interested in is, ultimately, I'm interested in evolution of species, and I'm arguing that you can change the problem a little bit by thinking about the evolution of cell types and breaking down the problem further than evolution of the whole genome sequence, but rather thinking about how did cell types change across time. And for me, what I'm interested in, when in evolutionary time did a particular cell type appear? Does the introduction of a new cell type mean a new organism? I don't know. Maybe not. But I'm very curious about whether this is possible. This is the kind of tree I'd like to build. So whereas comparative genomics can find the phylogenetic relationships of species, what I'm doing is using comparative single-cell transcriptomics to find phylogenetic relationships of cell types. For example, which cell types cannot change within the vertebrate lineage. Of all the cell types we have observed, of which we've probably not observed all, which cell types are consistent and appear in all organisms of a particular lineage? Which cell types are unique to humans, to zebrafish, to so on, many different species. So now we'll talk a little bit about the results, or the methods first. One question is, how can you map the cell labels, the cell annotations from one species to the next? Recently, there are some really great papers out publishing the morphogenesis or organogenesis of early animals, of animals in embryogenesis. So we have a great data set in mouse and a great data set in zebrafish. Now, if we wanted to map, like, where is this cell type in zebrafish land and where's the zebrafish cell type in mouse land, how would you do that exactly? Most of the cross-annotation data sets rely on one-to-one gene mapping. So integrating the data sets across multiple conditions. I didn't add your paper yet, sorry. I rely on one-to-one gene annotations across species. I think within species this is totally fine, but from the work my colleague has done, at least for within species, she really likes SCMap, because one of the things that's nice about it is that it allows for cells to be unlabeled or unknown between data sets. One thing I want to point out is that not all genes have a one-to-one match between species. So this is a gene called MITF, and in mammals, it has 3 alternative exons, alternative first exons. So that means that this transcript, because it appears as exon 1A, so each box is an exon, the line is the genome. The line lines in between these exons are introns that get cut out or spliced out in the final version. And what we can, what this is showing is that in mammals, we have an isoform that has exon 1A and then 2, 3, 4, 5, 6, 7, 8, 9, or 1B, 2, 3, 4, 5, 6, 7, 8, 9, 1M, 2, 3, 4, 5, 6, 7, 8, 9. Teleost fish, of which fugu or pufferfish is one, underwent a whole genome duplication in their lineage. And what's interesting is that many of the genes that in mammals are alternatively regulated by alternative splicing, so this could be argued as alternative transcription, are now regulated at the level of transcription absolutely. This transcription happens in a completely different place on a completely different chromosome. These are not one-to-one orthologs, right? This is 1 gene in mammals to 2 genes in fish, in fugu. And as I mentioned, most of these cross-species analyses use one-to-one orthologs. So if you look at the percentage of all human genes, just around 50% of them have a one-to-one ortholog in mice, and around 30% have a one-to-one ortholog in zebrafish. So you're certainly going to get a signal, and my argument is that you can get more. Now, single-cell RNA seq is pretty limited to annotated genomes. So given a tissue we're interested in, we do some sequencing, we get our cell types out of it. To get those cells out of it, we align reads from each cell to a genome. But if we don't have a genome to annotate to, we can't do this step. We can't align. And then we can't get our count matrix, which means we can't get our gene expression matrix. We can't do this clustering. We can't get this nice 2D plot that everyone has in their single cell paper. So the solution I propose is to use protein k-mers created by 6-frame translation of RNA k-mers. So a k-mer is a DNA word of length k that we chose. And by performing 6-frame translation, we're bypassing the orthology problem, as protein sequences are more stable across species than DNA sequence. And here we can also ignore, well, not so much ignore as bypass the issues with isoforms as well. If we're looking at all reads and sampling from all reads some k-mers, then we're now allowing ourselves to include all possible isoforms as well, and we're not collapsing to individual gene labels. So I will be using not the entire sequence, not the entire proteome of a particular length. I will be using a sketch. So a sketch, just like a drawing is a compressed representation of a person, for example, a sketch of sequences is also a compressed representation. So in this drawing, I mean, we as humans can see that this is a person with an arm and a hand. This arm is literally scribbles, right? And yet we can tell it's an arm. We don't really need to see all the folds of the fabric and so on. What's very interesting about about images, as humans, we now have been trained over much evolutionary time to identify faces very easily and identify other body parts easily. So in this sketch, we know this is a person. This is not a landscape. This is not a like still life painting. So as humans, we know which features to use. Unfortunately, in biology, I argue we don't know enough to know which sequences and which features are informative across all. So what we do instead is take a random sampling. And how do we do that? This example, I will caution, is with RNA k-mers or DNA k-mers. And I will show you at which point we do the protein translation. So given a sequence, we chop it up into k-mers, in this case of length 6. I realize that length 6 is not ideal for DNA because you can have a k-mer whose is equivalent to its reverse complement. So typically with DNA k-mers, you don't use an even number. We've chopped up our sequences into k-mers. Okay, after step 1, we translate to protein and then hash each k-mer into an integer. So hashing is a commonly used computer science algorithm. This is like what is behind Bitcoin and making sets equivalent. So you convert each k-mer into an integer. This hashing is random, pseudo-random. It's not cryptographically random. You can generate passwords with it that are 100% secure, but what you do get is a random ordering of all your k-mers. Like if you look at this, this is not ordered by their sequence. It's not A, C, A, T, or G is next in the alphabet, not T. So you have a random ordering of all your k-mers, and it's unique, right? Hashing is unique given the same input. You'll always get the same output. It's deterministic, given the same random seed. So in this case, we have chosen to observe a scale of 10, which means 1 out of 10 k-mers for the whole data set. Since we have about 20 k-mers in our data set, we're just retaining these 2. So from the original data, original longer sequence, we retain these 2 k-mers. And now our sketch of this sequence is these 2 k-mers, TGA, GAC, TGT, GAG. And as I mentioned, in step 1 is where we would convert to the protein k-mers. So at this point, I apologize, I have not made this slide yet. At this point is when we would still be hashing on the amino acid sequences, not on the DNA sequences. This triangle that no one ever understands is supposed to be sorting by the hash value, so from highest to lowest. So what we're interested in estimating is the cell-cell similarity. Given two cells, A and B here, how similar are they? Well, what we truly want is the Jacquard overlap. So that's how many Kmers overlap between one cell and the next. So that's from information theory. So A intersect B divided by the union of B. Or like, I like to call it the Venn diagram distance, but just what's the size of the Venn diagram here, or this overlap here. So if we have two cells A and B, and every little circle in A and B is a Kmer, the filled circles represent the Kmers that were actually observed, given our sketch. Now, when we hash them, we hash all our Kmers, we have some values, 42, 64, 82, so on, for A, sketch of A, sketch of B, and the sketch of A union B. So the sketch of A union B is a little interesting because notice it's the same length. In this case, we chose 5 Kmers for this. So we have the sketch of A union B. Because we're comparing small sets, we need to make sure our sets are of the same size when we're actually doing this comparison. So the total size, in this case, for the numerator here would be everything that was found in either A or B intersect by A and B. So we have just this one purple Kmer here with a cross. So this cross here is representing things that are in the sketch of A union B. In the end, our similarity of overlap here between A and B is the number of things that overlap in both A and B. So just this one hash, 82, divided by the total size of the hashes, which is 5. So our total amount is 1/5, or 0.2, between these two cells. A side note of what's interesting is that between two single cells, if you look at literally all of the Kmers that are shared, it's actually really small. It's somewhere around like 0.1 for similar cells, 0.01 for less similar cells. So for single cells, you really are undersampling the data a lot, which is interesting. So the data I'll be using as an example is from a recent paper that was single cell RNA seek data for about 100,000 cells in mouse. It was a sex-balanced study, sex-ish balanced, so 4 males, 3 females. And this was performed in both SmartSeq 2 library prep and a 10x Genomics library prep. So we were able to do quite a bit with the data and make it really available. I was part of the people who pushed, I was pretty much the only one who pushed for using a cell ontology or a controlled vocabulary of cell types, because it makes the data much more usable later on. And also all of the data is available on Figshare. So if you just want matrices and labels, you can get them. For some reason, that's really hard to find from other papers, and it's really frustrating. Today, I will be focusing on the kidney. I'm not a kidney expert, so the main things that I look at are, in this more complicated diagram, is that there's blood vessels, there's some tubules over here, and there's tubules over here. That's what I focus on. In tabula muris, we found about 5 cell types in the cells dissected from the kidney. Some immune cells, because there's blood and everything, and so that was part of what we found. Some tubule cells from over there, some tubule cells from over there, and endothelial cells or other cells lining the capillaries. So if we use a smaller Kmer size and build a nearest neighbor graph, so each dot here is a cell, and I've made an edge between cells if they were in the top 5 of their friends, if they're most similar to the other, in the top 5 similarity of each cell. And so the tighter that group is, because this is a force-directed spring layout, first the graph tries to pull apart as much as possible, but as these groups that are more similar group together, then we see this more tightness. So a very, very tight group here indicates that all of those cells are very similar. So if you use K equals 4 amino acids, you get what looks like if you threw confetti on the ground. Like you just have a bunch of cells all mixed together, all your cell types are mixed together, when in the previous data, we previously saw that by this traditional single cell RNA seq analysis, you saw these separating quite differently, quite nicely. So what you can do is increase the length of your protein chimer, so to 5 amino acids or 7 amino acids, and then within mouse, you start seeing more and more separation of the immune cell types from the endothelial cells and the epithelial cells. Now, as I mentioned, in teleost fishes, they had a slightly different biology, and in these fish, immune cells develop first in the kidney rather than the bone marrow. If you think about it, fish don't have very big bones. Whereas in mammals, the primary site of development for immune cells is in the bone marrow. So to this mouse kidney data set, we're going to add 2 more data sets. We're going to add a zebrafish kidney marrow data set, which could be similar to our original data set because it's kidney and it has some immune cells, some immune cells that could match up. We're also going to add a human bone marrow data set, which could be similar because there's immune cells or because it's mammalian. But really, these 2 data sets should be most similar since they're both immune cell development sites. So maybe, maybe not. If you use a small case size, then you get a bit of confetti. And as you increase your chimer size, the delineation between species becomes quite strong. And by the time you use K equals 7, as we saw, that was very useful for delineating cell types. Turns out it's also useful for delineating species. So one thing I am interested in doing next is to deplete the species-specific signal using commonly used machine learning algorithms in text processing. So when you're comparing documents, looking at words that appear often within one document. So removing the, a, and said, then, and so on. What's interesting is that the human and mouse data sets do still seem to overlap, and I will be looking into which cells exactly those are. If you do color by, is that a question? If you do color by cell label, then you do see that, yeah, within zebrafish, you see differences between cell types. But for human and mouse, you may not necessarily see quite yet all the similarities between cells that you had expected from the immune cell lineages. So going on to the future work, I will be, as I mentioned, I will be removing species-specific chimers with an algorithm called term frequency inverse document frequency, or TF-IDF, which is widely used in machine learning, deep learning for removing commonly used words and removing things that are adding similarity where we don't need it. So within a species, we don't need that similarity. I'm interested in taking the protein chimers that we have that are enriched in particular cell types and comparing that to what we have with bottom-up proteomics with trypsinized proteins. Since a lot of data sets that we have are stranded RNA-seq data, we can also do just 3-frame translation. We don't have to do 6-frame translation as we would if we had just DNA. And we'll also need to compare the same data sets to the nearest neighbor graphs and other processes built on the gene counts of 1-to-1 orthologs, since that's the standard of care for this data at this point. I wanted to finish up with my acknowledgments to people at BioHub and outside of BioHub. Outside of BioHub includes SourMash, which is the program I use to generate the K-mer signatures, which led me to Bamnostic Marcus's tool because I needed to add functionality to read 10x genomics spam files. Nextflow, which is my current favorite workflow engine. Reflow, which was my previous one, which is not my current favorite anymore. And the entire data sciences team for being really supportive. So that I want to take any questions.
Hi, I have a question.
Can you say your name?
Yes, sorry. My name is Yiwei. I'm one of Josh's master's students. So I have a question about like doing the step of translating the RNA K-mer into protein K-mer, because you mentioned one of the reasons was because the protein sequence is more stable across the species. I'm just wondering if all the like RNA sequences, are they like in reality on being translated into protein? If they're not, then doing that, how would that help you like the stable part of it and do the mean hash and do similarity?
Yeah, that's a great question about the non-coding RNAs. Like, what if your RNA does not end up in a protein product, but rather as a non-coding RNA? Why do the translation? Because in that case, yeah, the nucleotide sequence would be a lot more conserved there. I think when you're translating to protein, yes, I'm ignoring non-coding RNAs. I would argue that since we are translating to protein, that if the nucleotide sequence is conserved as a non-coding RNA, yes, it doesn't represent itself in that's not a true protein product that we're looking at, but that sequence is still going to be conserved, because if it was TTT in mouse, and for that function of the non-coding RNA to also have to be TTT in human, then those protein sequences, no matter how you translate them, will also be similar. Thank you.
Hi, thank you for your talk. I think this is a very brilliant idea of using K-mer rather than mapped RNA seq data to quantify these cell types. So my question is, it seems that there is assumption in this analysis that this Jacquard distance of the protein K-mer similarity somehow can reflect the evolutionary relationship between different cell types. I was wondering if there is any underlying justification for that. Thanks.
Yeah, so let me go back to this slide. So in thinking about the evolution of cell types, the way you bring up a good point that the way we can obtain these cell type labels can be through protein similarities, but obtaining this tree may not necessarily be reflect the protein changes. To obtain the phylogenetic tree, that's when we need to go back to the DNA or RNA sequence to see how the RNA sequence of the cell types has changed to get the evolutionary tree. But to get buckets of cells, that's what we use the proteins for. To get the tree, that's what we use the nucleotide sequence for.
When you're developing the sketch that you're going to be passing and matching your K-mers to, how does that control? Because again, you said that the sketch is defined by the hashes that are completely randomized. It's not based off of the actual sequences that are in there. How do you prevent it from grabbing duplicate regions or repetitive regions that you may randomly select it based off of its hash value, but because they're randomized, you can't tell if the two k-mers are the same exact sequence.
Okay, so I think if the k-mers are the same sequence, then their hashes will be the same. So what's nice about hashing with a particular deterministic algorithm is that given the same input, that you're uniquifying your data set by hashing, which is nice. One thing that can be difficult is that if you're looking for k-mers that appear in low abundance transcripts or that like never really appear in your hashes, there's other algorithms that have appeared recently called a universal k-mer set that allows you to guarantee, given a particular length, say your read length, that you have a k-mer sampled from each read.
Okay. I thought that you were saying that any k-mer was just given a random hash, not that the hash was actually developed off of the k-mer.
Yeah.
Now that makes sense.
Got it. He had his hand up. Oh, I'm going to answer his question because he had his hand up.
So then going off of that question about the sketch, so the few k-mers that you're selecting out of the sketch just seem to be based on the lowest hash value. Am I right about that?
Yep, it's called a min hash for that reason.
Okay. Is there any way that you could potentially like sort these k-mers based on how much information, how characteristic they are of the RNAs, so that you could pick out something that has a lot of information to it in a small amount of data, rather than just randomly pick a couple k-mers that might correspond to like repetitive regions or something like that?
Yeah, there's a lot of ways to pick k-mers. I'll say that. There's a lot of ways to pick k-mers. I think, so this universal k-mer hash thing is something I'm going to be moving towards. There's another method called hyperlog log or HLL that another team uses that does a slightly different kind of hashing and uses a few less bits of information. But yeah, one of the problems is how representative, how do you know the k-mers you're picking are actually best for your data set? And short of filtering your data through like gene transcripts that you're interested in, I don't know if there's a great way that isn't introducing biases that we already have about the data, about what genes we've already seen, what genes have been sequenced already, things that I would like to sidestep, especially moving towards non-model organisms. Yes, now I can get to your question.
No, that's fine. I need eyes to back. I'm wondering about the context.
Yeah, so calculating a sketch is takes seconds. Like that's not a long time. Calculating thousands of sketches across many different protein translations for 50,000 cells takes a long time. So what then? I think as people writing tools, I think we have a responsibility to write them in a way that is parallelizable. It's like I use both local compute on-prem, as it's called in the like corporate world, as well as in the cloud for using AWS. AWS is nice because then I can deploy, my like 2,000 cell, 5 different k-mer sizes, 2 different molecules, DNA, protein, and then like 6 different sketch sizes. So however many 100,000 of jobs that is, I can deploy pretty quickly using Nextflow, which is nice. But it does take time to compute. The main thing that it takes a lot of time is the comparison, like loading all the sketches in and then creating your cell-by-cell matrix of comparisons. I think what's nice about the sketches is that you can make on-the-fly databases, and using commonly used database techniques and binary search trees, you can create really robust search databases. So eventually, given all known cell types, given a database of all known cell types, it would be very easy to add one data set and say, okay, which one is which cell is closest, which cell types are closest. Give me the closest 10 or 20. Like that's probably plenty to understand what kind of cell type that is. That's where I want to move to, because the big matrix, when we're going to be getting to however many billions of single cells we sequence, that's not going to be tenable. Oh.
I wanted to ask about how expression level plays in here, right? So each k-mer is expressed.
Yeah.
How do you incorporate the number of copies of each k-mer in your sketch?
That's a good question. So abundance of the k-mers actually totally messes things up. Where'd it go? Too many slides. What was I thinking? So when you incorporate abundance of the k-mers, what's interesting is that when you're collapsing to genes, you're actually removing a lot of the noise that you would normally see if you only looked at sequences, because here it is. So this is a different data set. This is bladder, my favorite tissue from type Lammaris, because it's like 3 cell types and the people are really nice. And so if you look at just k-mer presence absence, so this is on a K of 27. This is actually at the DNA level or RNA level on the 10x genomics data. If you compare the binarized gene expression, so take that gene expression matrix and just like make it true false, like is it above 0 or not, then you can get somewhat of a messy graph, you could say, but still pretty representative of what the underlying data is. And this would be the graph if doing the traditional gene expression route. If we do a nearest neighbors graph on the k-mer abundances, so this is not a Descartes distance this time. This is actually a cosine distance. So this is the angle between 2 vectors on their abundances for a particular k-mer. Then you get something more like confetti. Like maybe you organize your confetti a little bit, but still your cells that shouldn't be similar to each other are. I think the reason for this is because the coverage of single Saw RNA Seek is so sparse. So for one cell, you may have lots of coverage here and some coverage here, maybe some coverage here, lots here. So even though this k-mer was observed in both cells, so say but this one it was 100 and this one it was 2. Right? So even though when you collapse to genes, let's say these both collapse to 102 in terms of the gene count, but because the coverage is so spiky, that same k-mer may or may not have a similar expression value for that particular location.
Great. Any other questions? If not, let's thank Olga again for a great talk.